is a Flores Eccles Presidential Endowed Chair in the Department of Molecular Pharmaceutics. He is also a professor of biomedical engineering and director of Utah Center for Nanomedicine at the University of Research at Utah. His research focuses on the design of polymeric biomaterials for localized drug delivery and understanding the relationship between physicochemical properties of inorganic nanoparticles with their toxicity. He published numerous papers exceeding 230 manuscripts and book chapters, 500 abstracts, and he's given over 265 invited talks. Dr. Ghandehari served as executive editor and editor-in-chief of Advanced Drug Delivery Reviews, the #1 cited journal in the pharmaceutical sciences category. He is a fellow of the American Institute for Medical and Biological Engineering, the American Association of Pharmaceutical Scientists, and Control Release Society. He received his bachelor's in pharmacy and PhD in pharmaceutics and pharmaceutical chemistry from the University of Utah. The floor is yours, sir.
Thank you so much, Dr. Dokmechi. It's an honor to be here or there and really give a talk to colleagues and friends of Terasaki Institute. I also would like to thank Dr. Khadim Hosseini to really lead this institute and also having me here today. What I'm going to talk about is some of the approaches in drug delivery that we're taking in our lab as soon as I can remove this ribbon from top of my screen. And so basically, of course, as you know, modern drug delivery is over 5 decades old now. The question becomes, what are the things that we could do to innovate in this space? There are several areas that we could work on. One of them is advances in the design and characterizations of materials. How can we try to get more detailed definition over the structure, polymers that are better defined, nanoparticles that are geometrically better defined? How can we control surface properties of materials that are used in drug delivery? Of course, as we know, traditionally, a lot of the polymeric materials that are used in this area are polydispersed. Can we try to come up with strategies to make monodispersed polymers that can have better biodistribution and cellular uptake properties? And also, in addition, what are some of the advanced characterization techniques that we could use to better understand the material that we're using in drug delivery in order to correlate that structure with function? So definitely advances in materials design and characterization is one area that we can innovate in drug delivery. The second is a better or detailed understanding of the biological fate of the materials, these new materials that we're making or other people are making. What is their biodistribution? How are they taken up by the cells? How are they trafficked into the cells? We need to know about their toxicity in order to be able to use them as useful materials for drug delivery, and of course, their degradation and elimination from the body. What are some methods that we could use to enhance delivery of bioactive agents? For example, this area of using or combining physical triggers or pharmacological agents to enhance transport across cells or delivery is an important one. And I'll try to give one example today that I think more and more we're seeing combination delivery approaches where you can use, again, a physical trigger as well as other types of delivery systems to improve delivery. One could argue perhaps that to advance drug delivery, we have done a lot of work as a community of drug delivery scientists over the past 3 or 4 decades to develop our own delivery system, but maybe not as much work has been done to better understand the target biology. Because as we know, for example, let's say if we take an example of tumor targeting, we know that the vascular physiology is dynamic. And by better understanding that target biology, we're able to design delivery systems or approaches that will take us to specific tissues or target cells and subcellular sites in the body. Obviously, we need to think about, and I know Terasaki Institute does a great job of that, as well as many other places in the country, that how do we try to clinically translate these systems from benchside to the bedside? And of course, in that case, sometimes it takes years for new materials to find its way into actually clinical use, but also can we use existing delivery systems for newer and better delivery approaches? And finally, use of bioinformatics and artificial intelligence, it's finding its way into basically advancing drug delivery systems, as many other speakers probably previously at the Institute have elaborated on. So those are some of the ways that we can innovate drug delivery systems. And briefly, what we do in our lab, we have a few projects going on along the similar lines, if you will, that is to try to improve drug delivery systems. One of the systems that we have been working on for several years is use of recombinant polymers for localized drug delivery as liquid embolics. I'll expand on this a little bit more, but basically what this recombinant polymer technology provides is the high degree of control over polymer sequence and length that will then translate to higher degree of control over delivery and elimination of these systems. I'll expand on that a little bit in my talk today. The other area that we work on is that we try to utilize the inflammation, if you will, the extravasation of blood vessels that are present in the inflammatory side of sinuses. So chronic rhinosinusitis is a condition, basically a chronic condition that people have inflammation in the sinonasal area. And we have tried to characterize the vessels in this site of inflammation, and we have shown that these vessels are fenestrated, and we have shown that using liposomal systems, actually you can enhance delivery of imaging agents as well as anti-inflammatory agents at this site. Today, I will not be able to, because of time limitations, to talk about that project. A third project that we work on in our lab is to try to understand what is the influence of factors such as size, shape, porosity, and surface properties of inorganic nanoparticles, in particular silicon nanoparticles, on things like cellular uptake, the pathways by which they're taken up, to what extent are they taken up, for example, by macrophages, and how would this influence on composition as well as size, porosity, and physicochemical properties influence their toxicity. So that's the so-called nanotoxicology project. The reason that is important is that if these particles are going to be used for biological applications, including drug delivery, you have to have a better sense of how these physical chemical factors influence their biological fate. Unfortunately, again, today I won't have time to talk about that. My talk is primarily focused on polymeric drug delivery systems. What I showed on the top left, I believe, is probably top right of the screen, the recombinant polymers. Primarily, we make hydrogels out of those, but one could also use linear water-soluble polymers for targeted delivery to sites of tumors. And I will show a couple of examples in our lab where we, in combination with physical trigger, in this case, hyperthermia, as well as targeting the extracellular matrix that we have been recently focused on. And finally, a more recent project in our lab involves strategies to reduce absorption of drug molecules, and specific hydrophobic drug molecules, to this so-called extracorporeal membrane oxygenation systems, or ECMO. ECMO systems are used for critically ill patients that their blood needs oxygenation. Oftentimes, these patients undergo anesthesia, or they have to, the physician has to administer a number of different drugs. The challenge is that these ECMO systems are manufactured by different companies. And in addition, the plethora of drugs that are administered to the patients are absorbed to different extent to these ECMO systems. And the idea is either you can modify the surface of ECMO tubings, inner tubing, so that you reduce the absorption. That's one way to do it. The other way, the idea would be, can we modify drugs to reduce this absorption so that the clinician would have a better handle over dosing. So those are the three projects that I will try to talk about today. So in terms of the utility of recombinant polymers, this technology is a few decades old, so it's not necessarily new in our lab. In fact, the first report is from Joe Capello that he reported in Biotechnology Progress back in 1990, the systematic production of these polymers by recombinant techniques. But over the years, we have tried to utilize this technology for localized delivery applications. And the idea would be that with traditional polyamino acids, as you can see on the top of the screen, you have a random sequence of monomers. Let's say you have polylysine histidine. In random copolymers, you have lysine, histidine, histidine, lysine. You don't really have a controlled sequence of the amino acids. As a consequence, what you get is not a systematic structure-function relationship. Over the years, people such as Dan Urey and others, the late Dan Urey, they came up with strategies to make these polymeric systems by sequential polypeptide synthesis. In other words, you make a pentamer or a sequence that has 10 amino acids or what have you, and then you multimerize that chemically, or polymerize that, if you will, chemically. And in that case, you will get some degree of control over sequence of the monomer and the polymer sequence, but that degree of control is not long range. In other words, you cannot dictate the sequence of the polymers. Let's say I want to have, for whatever reason you might want to have, amino acid sequence RGD at sequence 101, 102, 103. It's impossible to do that with this technology. In addition, you still get a polydispersed system, and that's one of the challenges with this sequential polypeptides. So with recombinant technologies, you can get ideas from nature. This is shown on this side of the screen. You can get ideas from nature. Then you can try to basically backtrack the genetic codon for those types of sequences. And then you clone and express. You multimerize the gene segments, you clone and express, and you produce protein polymers that are monodispersed. You have long-range control over the sequence. I'll show you one example later where we have put MMP-responsive sequences at strategic locations in the polymeric backbone. And as a consequence, you can have a better control of the structure, which leads to better control of the function of these materials. So one of these polymeric materials that we have worked on, which I will show you the structure in a second, are the so-called silk elastin block copolymers. So if you could imagine, the blue segments here are the elastin portions. The red segments are the silk portions. As you increase the temperature from, let's say, room temperature to body temperature, these elastin blocks collapse. They bring the silk units closer together. They hydrogen bond, and they form hydrogel material. So this is shown, for example, as you deliver these polymers at higher temperature, they start to form a turbid solution. And in the next iteration, you can see that you can nicely get hydrogels out of these systems, depending on the number of silk and number of elastin that you have in the monomer sequence, but also importantly, depending not only on the number, but also the exact sequence of silk elastin that you have in these polymeric materials. So over the years, we have synthesized or biosynthesized, if you will, a number of these analogs. Several of them are showed here. In brackets, you can see the monomer sequence. And in this monomer sequence, for example, you can see that we have 4 elastin units, 1 elastin unit that has a lysine residue, and another elastin unit that is repeated 3 times, plus the silk units that's repeated 4 times. And the entire sequence, monomer sequence, is repeated 12 times, plus some head and tail sequences. So in this case, we call this 47K, where you have basically 4 silk, 7 elastin, plus 1 elastin that contains a lysine residue. We have systematically changed the length of these elastin and silks, and basically we came up with 3 structures, the first generation, which are 47K 415 with a longer elastin unit, and 815 with both longer silk and longer elastin. And then we looked at the modulus of elasticity of these polymers. You can clearly see that silk 47K that has higher ratio, but also the specific sequence of silk and elastin has higher modulus. The 815 is somewhere in the middle, and the 415, which has long elastin units and shorter hydrogen bonded crosslinks, has lower modulus of elasticity. The idea is that you can control the modulus, the stiffness of these polymers, by controlling the sequence, but you can also control the degree of swelling. These are after gel formation. You can see that, for example, 815 that has longer elastin units over a period of time actually shows sensitivity to temperature. And we did some basically SANS at NIST a number of years ago to look at the porosity of these polymers. One of the areas that we have used these polymeric materials is for intratumoral gene delivery. And in this case, what is shown here basically we incorporated adenoviruses in these liquid polymers, and then we injected into the head and neck tumors of basically xenografts of mice. And the key point here is, as a function of the structure of the polymer, as well as function of concentration of the polymer, we were able to control the release of these adenoviruses and gene expression. The bottom line is that everything else being equal, not only polymer concentration, but importantly, the structure of the polymer has a profound influence on how much of these adenoviruses are released and provide gene expression over a period of time. The reason that you see this decreased expression doesn't really have to do anything with the polymer. Rather, it has to do with the fact that the viruses lose viability over a period of time. But the take-home message is that by controlling the structure of the polymer, we were able to control how much of these genetic materials are released and lead to gene expression. If you were to use the same polymeric materials, but at lower concentrations that they don't form gels, and you mix them with adenoviruses, using atomic force microscopy, you can see that a lot of these viruses are really interacting with the polymers. And we think that this is in part the reason that these viruses are more stable over a period of time. And in part, that's the reason that they give more transfection efficiency. But the main reason actually is the fact that we can control the release of these materials, the viruses. One of the challenges that we had in those first generations, and I'm kind of fast forwarding, with the 815K, as you recall, was our lead polymer. And one of the challenges that we had, they didn't degrade fast enough in the tumor. Let's say if you want to deliver the material over a period of months, after a while, when we excised, we could easily excise the hydrogel from the tumor. And as a consequence, they were not degrading fast enough. So we were interested to design silk elastin block copolymers that would degrade in the tumor environment. And for doing so, we incorporated this green sequence, which is basically a sequence that's responsive to matrix metalloproteinases. Many tumors, as you're aware, they overexpress matrix metalloproteinases. That's in part the reason that basically tumor cells go elsewhere and metastasize. And so these enzymes are overexpressed in the tumor. In this case, we were interested to use this property of MMP responsive sequences to incorporate it into the polymer backbone in order to facilitate the degradation of these materials and hopefully enhance gene expression as well, due to the faster release of these viruses. So what you see on the lower hand side, so we spent some time, we made one prototype and later a couple of other prototypes where we could precisely control the location of these MMP responsive sequences in the polymer backbone. What you're seeing on the lower hand side, let's say the total protein loss, which is protein that is resulting from the degradation of these protein-based polymers. On the left-hand side, this is SELP815K. This is the parent polymer that doesn't have the MMP responsive sequence. You see certain amount of protein loss, but when we incorporate in the presence of two different types of enzymes, MMPs, but when we incorporate the MMP responsive sequence, you see increase in release of the protein, which is basically increased degradation of the material. This degradation results in lower compressive modulus because the polymer system, the hydrogel system, is sort of falling apart. And so you get less network density. And on the right-hand side, first we looked at the release of certain fluorescent beads to see whether we can enhance the release of these fluorescent beads that are roughly the size of viruses. And indeed, we could see the increased release of these fluorescent beads. So this first prototype of MMP responsive silk elastin block copolymer worked. And so the next thing that we did, again, I'm fast forwarding a lot of synthesis, characterization, in vitro evaluation. But what you're seeing here is basically the delivery of an adenovirus that contains the thymidine kinase gene, which also has a luciferase gene to enable us to visualize. In certain types of head and neck tumor xenograft models, basically the viruses were mixed with the polymers, they were injected in the tumor, and we looked at the percent survival of the animals. So let me explain what types of polymers did we use. If you recall, this 815K is the parent polymer that gave us the highest transfection efficiency. Then we also synthesized a polymer backbone that has, we call it RS1, where the MMP responsive sequence is exactly between the elastin and the silk units. And then we made another type of polymer where the MMP responsive sequence, we called it RS2, is in the middle of elastin block. And then the third type that we made this MMP responsive sequence within the silk unit. With traditional polymerization techniques, it's impossible to make such a long polymer of somewhere around 70 kilodalton, where you can precisely control monomer sequence and polymer sequence. So in this case, we did that, and then we mixed these polymers with the adenoviruses that I just mentioned, the therapeutic adenoviruses, intratumorally, and we looked at the percent survival of the animal. And what you're really seeing, bottom line, is that in one of these polymer cases, everything else being equal, meaning the concentration of the virus is equal, concentrations of the polymers, in this case, equal, we get survival up to 50 days, which is that's as long as we did these studies. And this demonstrates the influence of the structure of the polymer on the efficiency and activity of these types of adenoviral delivery systems. So the reason we believe for that is that if you look at, but then we excise the tumor, if you look at SELP815K, we have film formation, it doesn't quite degrade. That is also true when we did in vitro degradation studies. But when we incorporated the RS responsive sequence within 815K, we could see that the polymer is degrading in response to MMP, and basically the viruses are penetrating, and it gives us better activity. Really, bottom line I want to emphasize is I think the crucial importance of control over structure that gives you this kind of nice biological activity compared to those other prototypes that we made. Another area that we have relatively recently started to try to explore these recombinant silk elastin block copolymers, to use them as for embolization. In this case, for example, we're trying to use these silk elastin-like block copolymers as injectable embolics for transarterial chemoembolization. Basically, if the patient has liver cancer, you've got vascularity, and a lot of people try to use this vascularity to deliver things like lipiodol, which is an oily material mixed with drugs, for example, sorafenib, or people have developed beads that will be delivered through the femoral artery to the liver tumor, and then these beads are going to occlude the blood supply. What we have hypothesized is that our protein-based polymer can easily be dissolved in aqueous environments, so you don't have any residual solvents. In addition, if we can control this, we hypothesize that if we can control the structure of the polymer, combination of both the structure and concentration of the polymer will give us a polymer that will gel around the solid tumors in these microvessels. It will penetrate further, gel around it, and then basically kill the tumor. That's the idea. So the design parameters that we needed was to have a viscosity not more than 150 centipoise, so we can easily inject these polymeric materials until they reach the tumor. We wanted a gelation time less than 5 minutes, so we wouldn't have the intraarterial embolization, so we don't have any venous washout. We needed a stiffness, if you will, of these polymers of 1 × 10⁵ Pascal, so it withstands the hepatic arterial pressure. And optionally, in the future, we would want to have a system that we could easily incorporate bioactive agents into these polymeric materials and basically try to deliver them as chemoembolic. So that's the idea, would be delivered through this catheter. Optionally, you can deliver imaging agents, what have you. Some of the in vitro studies that we did, we needed to make sure that we reached the viscosity that we needed to reach, and SELP815K gave us that at 12% weight. Shearing of these polymers were extremely important because the silk blocks actually interact with each other, and if you shear them, you get this linearity of the polymers that enables the interactions of the silk blocks in a better fashion, so that shearing proved to be very important. This is non-shear, doesn't give us that viscosity, but the sheared polymers does give us. the modulus of elasticity, we reached the limit that we wanted to. And also, this is a non-tumor bearing rabbits that we demonstrated that we can occlude some blood vessels here. And in fact, later when we did histology, in the saline control, the arterioles are open. In the self control, we can see that we can clearly see that these selves are forming a gel. And what you see here is before we do the in vivo studies, we did some in vitro microfluidic examination of these systems where you deliver the dye, the red dye from here, the other side you deliver the polymer, they form the gel. And basically that demonstrates proof of concept in vitro and in non-tumor bearing rabbits in vivo that the system works. The next thing we did, this work we did with a contract research organization in porcine kidney to demonstrate the embolization of cranial pole of porcine kidney. What you see on the top left, or maybe it's your top right, is basically pre-embolization. You can clearly see the vascularity, post-embolization, and after 1 week post-embolization, you can see that the tissues are basically dying, causing tissue necrosis as a result of this embolization, that you don't see actually blood flowing through those. This is also true. The same is shown into these figures down here, where you have non-embolized control, 1 week post-embolization, and we could actually isolate the silk elastin block copolymers that have formed hydrogels in this kidney. If you look at histology, we can see beginning of re-endothelialization, which in some cases for as embolics, that's an important issue for the tissue to grow back. We are seeing the signs of it, and also we are seeing that with the blue arrows, they show the SELP 815K emboli clearly, and of course you see some macrophage infiltration. And what you see on the right-hand side is basically, as I mentioned, tissue necrosis as a function of embolization. So this is our proof of concept so far in animal models that these liquid embolics work. Of course, we have to optimize it, and we're in the process of trying to hopefully establish an appropriate liver tumor model in our lab, or at least collaborate with those who have it, so that we can demonstrate that this concept works as an embolic first, as a bland embolic first, and later as a chemoembolic for treatment of liver cancer. Similar concept can be used for brain aneurysm. So in this case, we hypothesize, of course, brain aneurysm, you have these basically bulgy, this bulge in the blood vessels in the brain. And so it's a very important issue. This actually was taken by permission of the family of this particular patient in Utah that had brain aneurysm, and it's a very serious condition. The current technologies that are used to address brain aneurysm are either surgical clipping. So basically you kind of, you seal the aneurysm completely, but you have damaging the surrounding tissue. It can leave residual material behind permanently. Another option is this mechanical embolics that are minimally invasive, but the problem is they require chronic anticoagulation. In many cases, they fail and they leave residual materials. Liquid embolics have been developed. They're minimally invasive. They have lower risk of failure compared to the other two. But the current liquid embolic, to the best of our knowledge, they use toxic organic solvents, and they can leave residual materials behind permanently. So we hypothesized, can we use our basically protein-based, aqueous-based liquid embolics where we have control over polymer structure and try to use them for basically brain aneurysm. So in this case, sorry if I'm just trying to use my ruler here. Sometimes it's difficult to move it around. Sorry about that. So in this case, we hypothesize that our cells are chemically compatible with all catheters. You don't have any absorption or adverse effects to the catheters. They gel without drastic change in volume. That's extremely important. As those of colleagues that are familiar with hydrogels, you know that a lot of times when you try to form gels, they have drastic changes, either increase or decrease in change in the volume, which would be not so beneficial. But these silk elastin block copolymers, the silks crystallize And once they form gel, they hardly change in volume. And in addition, we don't have any addition of organic solvents to the reaction. So the idea would be you basically insert a balloon catheter, then you insert, then of course you inflate it, then you insert the catheter that you need to deliver the liquid embolics, and then basically that's the general idea, that you will get embolization at the site of aneurysm, then you can remove the catheter, and that should do the job. Of course, it's very easy to illustrate these things. We had to come up with strategies to obviously make these polymers, but also demonstrate that they work. This is an in vitro. This is basically a tubing. The green stuff is when we have cell mixed with some green dye, and this is basically some red dye that is flowing through the tubing, demonstrating in vitro that the concept works. The next logical step was to do animal studies, and in this case, we used the carotid artery aneurysm model. Again, this is through a contract research organization. This is the type of the site of aneurysm that are developed by our contract research organization that develops the animal models. Then the idea would be, again, you have the balloon catheter, you have the guide wire, and try to basically demonstrate that after delivery of these liquid silk elastin block copolymers, you completely occlude the aneurysm. The video that I'm going to show you, I think it's going to clearly show this, that in the left-hand side, as you see, you have pre-embolization, you have the site of aneurysm where I'm showing it right here. It's clearly visible. On the right-hand side, you can see after you deliver the cell, then you don't see the aneurysm that you see here. You don't see that anymore over here. So again, the proof of concept that the same technology can be used for brain aneurysm as well. So the other area that we're using these silk elastin block copolymers is try to deliver radioprotective agents for basically prevention or treatment and prevention of radiation-induced proctitis. For many types of pelvic cancers, for example, prostate cancer, oftentimes, of course, there are several strategies that are used. One of those strategies is radiation of the prostate tumors. Despite the fact that basically these radiation systems, there's a lot of improvement in them nowadays, they can do a really good targeting. Still, patients suffer from radiation-induced proctitis because basically you're radiating the adjacent tissues. And so my colleague, Sai Uttamasetian, who works at Boston Children's currently, he used to be at University of Utah. He's a pediatric urologist, but also is interested in this topic. And so one of the drugs that we are using are these semi-synthetic glycan ethers that we call them GAGs, glycosaminoglycans, that he has shown that it has anti-inflammatory properties and effective in treating radiation-induced proctitis. The challenge that he has is that these drugs do not have the residence times that he needs them in the rectal tissue in order for them to stay around and try to prevent damage from radiation. So what we did, basically, we mixed our silk elastin block copolymers with these GAGs. We looked at their viscosity, we looked at the storage modulus, and we came up with a specific sequence of these polymers. I'll show it to you later, which is 415, that when mixed with these GAGs and delivered rectally, it has enough residence time to reduce radiation-induced proctitis. That's the general idea, and we demonstrated that by histology. But if you look at the next slide, basically, again, goes back to the concept of structure-function relationships. The 415K, the one with 4 silk and 15 elastin, gave us the best localization, if you will, in the rectal tissue, and gave us the best reduction in response rate. These are basically animal tests that we do. We look at pain response, and pain response is a good indication of radioprotection. And what you're seeing here is that with the combination of the drug with this polymer, particular 415K, which is shown to be localizing in the rectal tissue to a higher extent compared to these other systems. It gives us the best results of protection. One of the things that we have been wanting to do, but we haven't been successful, is to make nanoparticles out of these silk elastin block copolymers. We tried many different methods, and one of the reasons, as I mentioned earlier in my talk, some of the newer characterization techniques can give us an idea about the three-dimensional network properties of these polymers is liquid cell transmission electron microscopy. So we tried to make nanoparticles. One of my students made these nanoparticles from cells, but the challenge was that over a period of time, they would make these fibers. And the reason for making these fibers, and you can clearly see in this video that basically as a function of at 37 degrees, these materials, even though we made nanoparticles at the beginning, later they start to make fibers. And the reason they make fibers, we hypothesize at least, is that you still have silk units that are sort of protruding from these nanoparticles that interact with other silk units of other nanoparticles. And over a period of time, these polymer strands interact with each other and lead to the formation of these nanofibers. So I'm going to switch gear from gels and nanoparticles to water-soluble polymers. Many of you are familiar with this whole field that was actually coined by Ruth Duncan back then at Wells University and afterwards at Cardiff, this whole field of polymer therapeutics. You got polymer micelles, polymer drug conjugate, polyplexes, and what have you. One of these polymers, so-called polymer therapeutics, are these water-soluble polymer drug conjugates that actually Henry Kopacek in our department, back when he was in Czechoslovakia, pioneered this whole field to try to attach drug molecules to the side chains of polymers, optionally attach targeting moieties, and try to deliver them to the sites of tumor. So this concept is about 3 or 4 decades old now. And to this date, we don't really have a polymer drug conjugate that's clinically viable. One of the reasons, there are many reasons, we don't have enough time to really go over the field and say why they haven't worked. But one of the reasons is that because of their molecular weight, you have to control them such that they're excreted. Because of that relatively low molecular weight, let's say 40 kilodalton, they're not retained in the tumor as long as they should. So then we ask the question, can we combine physical triggers, such as physical hyperthermia, to try to enhance the delivery of polymer therapy? So the hypothesis is that if we deliver gold nanorods to solid tumors, prostate tumors, first, and then we deliver our polymer drug peptide conjugate, a peptide that recognizes heat shock proteins that are overexpressed as a function of increasing temperature on the surface of cancer cells, then because of hyperthermia, we're going to be able to increase not only the localization, but also the uptake by the cells of these polymer therapeutics. That's really the bottom line hypothesis. I know this is a busy slide, but I'll try to kind of walk you through it. What you see on the top left where my mouse arrow shows is that we delivered with this method polymer gadolinium conjugate. Right-hand side, this is a mouse with 2 tumors on the side. Right-hand side, you have basically hyperthermia. Left-hand side, you don't have any hyperthermia. And you can clearly see that these polymer gadolinium conjugates that don't have any targeting moiety, as a function of hyperthermia, we're talking about 37 to 42 degrees, increase the localization. That's number one. So even if you don't have a targeting moiety, you're going to get increased localization because you increase the vascular perfusion as a function of hyperthermia. Then we made a series of different polymer drug conjugates. These drugs are not attached to the same polymer, but to different polymers. And we found a lead conjugate that in vitro shows synergism as a function of basically combining hyperthermia and this polymer drug conjugate. When you attach this heat shock targeting sequence, you improve synergistically the delivery of these polymer peptide conjugates. And the reason is that these heat shock protein are basically overexpressed on the surface of prostate cancer cells in vitro. As a consequence, you get improved recognition and uptake of these polymer drug conjugates. Therefore, you see synergism. What you see on the lower left-hand side is that this is the tumor AUC area under the curve in the presence of hyperthermia with polymeric system that doesn't have a targeting moiety, that's untargeted. Just the hyperthermia by itself improves the area under the curve, delivery of this polymeric system. When you have the peptide, it further increases the area under the curve, as you can see here, but also it sustains the delivery over a period of time because of this increased expression of heat shock protein receptors and the fact that basically these polymer peptide drug conjugates stick to the site of action. And what you see here is basically in vivo, we demonstrated that when you have the targeting moiety plus the hyperthermia, you can improve efficacy in these systems. Another area that we have recently tried to show the utility of polymer drug conjugates is in extracellular matrix targeting. What are the advantages, or what are the challenges actually for delivery of drugs to tumor cells? Obviously, you don't get enough penetration into solid tumors. I showed that with hyperthermia, slightly we can increase that penetration. The late Dr. Maeda showed it with other types of things, such as, for example, incorporation of nitroglycerin in the formulation and other works elsewhere. But it's always challenging to deliver. And of course, we know that EPR effect really varies from patient to patient, varies from tumor to tumor. It's not a universal phenomenon. In certain cases, it doesn't work. In some cases, it works. But we have hypothesized that by delivery of drugs to extracellular matrix, you can circumvent that sort of penetration issue. Tumors undergo changes, and because of that, you have this basically changes in extracellular matrix. And the underlying molecular changes in extracellular matrix is that you have this denaturation, if you will, of the collagen that surrounds solid tumor. And so we asked the question, can we utilize this denaturation of collagen matrix around the extracellular matrix to try to target polymer therapeutics to that site? Of course, we haven't attached a drug yet, but what we have done is that, and I will show you the structure of these peptides in a second, we utilized certain peptide sequences that recognize denatured collagen in the tumor ECM and hypothesized that if you attach these to the polymeric systems, you can alter pharmacokinetics. This is known in the literature. You can sustain localization, and optionally, you can attach drugs and imaging agents, and you will have multivalency. So again, I'm fast forwarding. What I'm showing here is there are certain peptide sequences that were developed by my colleague, Dr. Yu, in our Department of Biomedical Engineering that target the denatured collagen. And what you're seeing is the water-soluble polymer, in this case, N2-hydroxypropyl methacrylamide copolymer, to the side chains, these collagen hybridizing peptides are attached. This is the scrambled, and this is the CHP by itself. So you can clearly see with the HPMA CHP, you get higher area under the curve of these polymer conjugates, demonstrates the proof of concept, so to speak, in osteosarcoma tumor models that you can get increased accumulation. And later, we have shown this with different types of tumors. We showed that with this ECM targeting, we can get increased breast tumor localization in osteosarcoma that I mentioned, and in pancreatic ductal adenocarcinoma. The next logical step in this project is to try to utilize ECM-targeted drugs and attach them to these polymeric materials to demonstrate increased efficacy. The last portion of my talk has to do with polymeric materials that we are trying to use to reduce drug. Sorry about that. Let me just go back. The last portion of my talk has to do with materials that reduce absorption to extracorporeal membrane oxygenation systems. These ECMO systems that are connected to critically ill patients, and they're oxygenated. As I mentioned, for clinicians, it's difficulty to dose appropriately these patients because there's variations in absorption of drug molecules to these systems. And because different hospitals use different types of systems, even coating the surface is not going to be a universal problem to address. So what we have done, we have hypothesized that drugs such as propofol, which is highly hydrophobic and is used a lot for ECMO patients, we can incorporate them into block copolymers of fluoronics, and then by having a hydrophilic shell around them, we can reduce the absorption. This is one of the very first studies that we have done in an ECMO circuit, not with a patient, but in an ECMO circuit. And we have demonstrated to some extent that this absorption is reduced. But now we have to optimize this, and we are using different strategies to hopefully get this to the level that we desire, so that it doesn't absorb as much compared to, let's say, Diprivan, which is the actual formulation that they're using. So we have been successful to reduce this absorption, but the next step is to really try to improve our system so that we could basically, hopefully, eliminate or at least reduce this absorption. One of the challenges that we have had in this project is that those ECMO circuits, each time we do these experiments, it costs us about $3,000. So with some of the undergraduate biomedical engineering students in our lab, we kind of have started to make ECMO mimics. We have made actually by now 6 generations, and I won't bore you with all the details. We changed, of course, we tried to control the temperature around this jacket. This is our ECMO mimic. This is an actual ECMO that my colleague, Dr. Kevin Watt in pediatric pharmacology uses, that I showed the experiments. But we have tried to make these ECMO mimics. So we have ordered all the tubings that are used in a real ECMO, but we have put it in a PVC device, so that PVC jacket plus temperature control. We found that temperature has a huge effect, and we made these different generations. And just recently, with our generation 6, we have shown that we can show the same level of absorption of propofol as you would see in an ECMO system. So what you see with these triangles versus the circle, this is the actual absorption of propofol in an ECMO system. This is the absorption of propofol in our ECMO mimic system. So we have been successful to make a relatively cheap, very cheap actually, device that we don't have to do all our drug screening with the actual ECMO. And once we do our screening with our ECMO mimic, then we're going to do our future studies. And we're trying to make, actually, as I mentioned, additional permutations of not only fluoronics, but other types of polymeric material to see how we can actually reduce this absorption. This basically shows the correlation of our ECMO mimic with our gold standard ECMO system, ex vivo ECMO system that is used. With that, I'll stop here. I would like to thank my research group. We had the honor of having Dr. Khadem Hosseini at the University of Utah. I hope we can see more of colleagues from Terasaki Institute at the University of Utah, as well as colleagues who are listening to this talk online. This is our research group in front of our building, and funding was from various sources depending on the project. This is the College of Pharmacy, a joint appointment between College of Pharmacy and Biomedical Engineering. So I go back and forth between, and these days I can say the weather is almost as nice as LA, but this is not typically the case. Thank you so much, Mehmet, for hosting me.
Thank you for this talk, Professor Hamid. I have a couple of questions. One is, when you do this drug delivery, is it based on the targeted? Can you elaborate on the targeted aspects of the drug delivery?
Sure. So I think we talked about 2 aspects, if you recall. One was localized delivery, when we have liquid polymers, they form gels inside the body. The other one that I showed with the hyperthermia, we have actually used the targeting moiety on the side chain that basically targets heat shock proteins in prostate cancer cells. And the other one was ECM targeting of the extracellular matrix as opposed to actual cell by itself.
Thank you. Are you also working on smart delivery system where I've seen results? I mean, you did extensive other people's work as well, comprehensive work, based on the need for insulin or some other problem that you
Sure.
deliver drugs?
That's a good question. In that sense, smart delivery systems, as you know, and I think as the late Alan Hoffman has described, has really a broad definition. One could argue that temperature-sensitive polymers are smart delivery, but I think what you're referring to is that delivering drugs in response to a physiological stimuli, such as increased glucose. We're not doing that type of work in our lab, but definitely that's something that obviously is of interest to a lot of drug delivery colleagues.
Thank you. What are the kinds of peptides selected into the hydrogel?
So the hydrogel that I showed is basically primarily three classes of peptides, if you will. One were the elastin units, that as you increase the temperature, water of hydrophobic hydration around the elastin units goes towards disorder and then collapses the elastin units. Then the silk units that help the formation of hydrogen bonds, and I showed the MMP responsive sequences for the hydrogel work. For the polymer work, for the water-soluble polymer, we had these heat shock protein recognizing peptides that would attach to the side chain, as well as the ECM targeting, the collagen mimetic targeting peptides that were in the side chains of the polymers.
Thank you. Another set of questions came in. Silk and silk derivative materials sometimes show immune responses. Do you see any evidence of immune activities associated with silk elastin-like protein products in your research results?
Yes, this is a very good question. Not only with silk elastin block copolymers, but always a major challenge with any type of protein-based materials when you deliver them systemically is the immune response. We haven't done extensive immunotoxicity on these systems. Having said that, we have done one study when we compared pluronics when they are mixed with the viruses and delivered intratumorally versus our silk elastin block copolymers. And in that case, we didn't see any difference in immunotoxicity. The second issue is that for the systems that we used, we have used them again intratumorally. So immune response would not be a major challenge. You get some absorption, but primarily these are going to be degraded and be eliminated. As far as systemic delivery systems, we haven't used them, but there are ways that you can overcome these. One would be by controlling the sequences or having sequences that are less immunogenic. The second one would be to have hydrophilic sequences that form hydrophilic shells around these polymeric materials. And some other groups have done that. But that's a very relevant question. And anytime these systems are to be advanced to the clinic, we need to look at that.
Thank you. One of the critical issues associated with aneurysms is inflammation. This inflammatory response can exacerbate the condition and potentially lead to further complications. So is it possible to simultaneously leverage SLP materials as a delivery system to administer anti-inflammatory agents at the site of the aneurysm?
That's a fabulous question, and the answer is affirmative, yes, because you can incorporate different types of drugs into these materials and basically deliver them, as long as they don't change drastically the viscosity of the polymer so that you get appropriate gelation. And even that, it's possible to control that because you can change concentration of the polymer when you mix it with a drug, anti-inflammatory drug, or if needed, you can change the structure of the polymer to give you the appropriate gelation kinetics when you mix it with a drug.
Thank you. What is the typical lifetime and biodistribution of its self nanoparticles in the body?
Self nanoparticles, as I mentioned, nanoparticles, not hydrogels. Those are two different things. Nanoparticles, as I mentioned, we haven't been to date, as I showed in that one slide, to really isolate self nanoparticles. Somehow, when we make them, these self strands that are protruding from the nanoparticles interact with each other. So I don't have the answer to that question because they haven't been made and they haven't been done. But drawing from what we know from the hydrogels, you can control the degradation of these systems theoretically if you can make them to a nice suspendable nanoparticle suspension, it can go from days to even months, depending on how much silk or elastin you incorporate to them. But we have not done that in our lab, and I don't have an answer to that.
Thank you. Thank you. This is, I guess, a futuristic question. Through which organs would these self nanoparticles eventually be cleared from the system?
Sure. If you make these nanoparticles, it wouldn't be any different than other types of nanoparticles. You get a lot of uptake by liver macrophages, by spleen macrophages, and potentially by lung, depending on how big these particles are and how they agglomerate. And the route of elimination would be eventual resorption of these amino acid-based polymers, and then basically elimination of the smaller amino acid motifs or the actual amino acids by themselves.
Thank you. Is it possible to synthesize self nanoparticles with different sizes and shapes?
Good question. It's possible. In fact, I think for example, some of the audience might be familiar with the print technology, which Dr. Joe DeSimone developed at the University of North Carolina, Chapel Hill a number of years ago. It's really elegant. He did a lot of work with PLGA. At some point, we actually considered, but that collaboration never materialized. But I think if you can come up with substructure that are basically don't adhere to these non-wetting surfaces that Dr. DeSimone had developed, it's possible to do that. That's one way that I can think of. The other way would be by bioprinting. It's conceivable to make these selves into kind of bioprinted to different shapes and things like that. And that would be really fascinating, actually.
Thank you. One of the limitations of micellar formulations is that it's not possible to filter or freeze them. Is it possible to filter through a 0.2 micron filter or freeze silk elastin-like protein nanoparticle forms?
Sorry, is it possible to freeze or filter or?
To filter or freeze.
Mm-hmm. If I understand, I hope I understand the question correctly, because we jump from, I guess, micellar to these types of systems, which are not micellar structures. Selves are not. Elastin-based materials can make micellar structures, but so far selves, they don't form micelles. But regardless of that, the answer is actually in the fermentation and purification process. When we make these, we kind of ferment these as kind of larger batches, and so we freeze them. We store them at 4 degrees centigrade. So in other words, we have to freeze them, and later we thaw them, and we use them for whatever purpose that we have. So it's definitely possible to freeze them. I hope I answered the question, if I understood it correctly.
I think you did. Another question is, what is the commercial status of these conjugated polymers? Are you thinking of?
That is a good question. I will refer the colleague who has asked this question to really some elegant reviews that have been written by Tuan Lammers at Aachen, as well as before that by Ruth Duncan a number of years ago. But basically, polymer therapeutics as a subfield, it has this definition. You could have bioactive polymers by themselves are polymer therapeutics. You've got micellar structures, polyplexes, polymer drug conjugates. And in those reviews, you can see a number of these are already commercialized. A number of them are in various phases, and a number of them had failed. And I think one of the best reviews that I've read is, like I said, is Ruth Duncan's, which really goes into the details of the influence of a spacer on the fact that they didn't finally make it to the clinic. And I can go on and on. I know we don't have time. In fact, I teach a whole lecture in my class on that topic. So a spacer can influence polymer backbone, length can have an influence, how the drug is released can influence, and a number of other factors. Cut the story short, to answer this question, a few of these systems, generally speaking, polymer therapeutics, have made it to the clinic, and a number of them have failed, and some of them are in various phases of clinical trials.
Thank you. Well, that's all the questions I have, Professor, and appreciate your time and willingness to give us a lecture.
I really appreciate the opportunity. Have a good one.
You too. Thank you.
Bye-bye.